All right, so here we have, obviously, the cortex. Now, you might be thinking, it's not obvious to me, and fair. So I'll show you this in a different kidney slide, but this is the one that has the good Macula Densa situation going on. The only place that you see renal corpuscles, which is, let's do yellow, that's more visible. So of course, this is a renal corpuscle, which includes the glomerulus, the podocytes, and the squamous cells lining the capsular space. You only see those in the renal cortex. So whether it's a cortical nephron or a juxtamedullary nephron, they both have their corpuscle situated in the cortex and not in the medulla. So if you look at a slice of kidney and you see this very distinctive shape, and you can even see it from low magnifications because you'll see the little crescent shape of the capsular space, you can be sure that you're looking at the cortex. Okay, yes, and it looks like some people are having an issue seeing the histology. All right, so as I was saying, the only place you see corpuscles is in the cortex, so it's a good visual indicator of where you're at in the kidney. Also, you'll notice that you're seeing a lot of cross-sectional views of tubes and not a whole lot in the way of straight longitudinal ones. That's another clue. So once you get into the medulla, which we will look at, the thing that you'll see is that the tubes are organized into longitudinal sections quite a bit in most cases. So presence of corpuscles, sections of tubes, those are your kind of main indicators for the slides that we have on Histology Guide. So the question that I was answering was about the juxtaglomerular apparatus and the macula densa. So that is visible in this picture. Let me zoom in on it a little bit, and I will help you sort of understand what we're looking at here. So every renal corpuscle has two ends or poles, kind of like the globe has a pole, right? So there's the north and the south one, which we arbitrarily decided based on magnetic stuff. We had to pick, so someone picked at some point. But regardless, it's an axis that goes through the center of a spherical structure, and where it pops out of that spherical structure is defining the endedness. So each corpuscle has, let's make this a little bigger, a vascular pole, which is the place where the afferent and efferent arterioles come and go, and nestled between the arterioles is this tube, which is just at the point where the DCT, the distal convoluted tubule, is coming up and transitioning to go try and find a collecting duct to connect to. So it's at that little transition point, but it's always going to be nestled between the afferent and efferent arterioles and be next to the cells of the juxtaglomerular apparatus for a variety of reasons. We'll discuss those. The other end of each corpuscle, which unfortunately you can't see here, it's really hard to get a slice that includes both, but I can show you the urinary pole in another spot. If it was present here, what you would see would be an opening like so, and then the tube immediately off of it would be the proximal convoluted tubule. So the urinary pole is the end of the corpuscle that the filtrate exits into once it's formed. So that's the terms that you'll see, and so I wanted to give you some context for that. So as you can see from this picture, let me just use this guy for a second. So you can obviously see some of the little arterioles of the glomerular capillary. So we can see one. Here's another one. You can see red blood cells here and there giving you little glimpses, but also remember that these are covered over by podocytes. So you're also seeing the nuclei and cytoplasm of a bunch of podocytes in addition to the capillary endothelial cells that are making up the capillary itself. So because of that, if I point to this structure, I wouldn't expect you to be able to tell me whether a given cell was a podocyte or a capillary endothelial cell, and I'm not going to. So because of that inability to visually make the distinction in this stain, if I point to this structure and I ask you what it is, glomerulus. We know it's in there. We can't necessarily distinguish it from the visceral layer of the capsule, which is the podocytes. So just go with glomerulus. That's all you need. Okay, so let's label some other things. We have got this little region of tubing. Now, here's a thing. Some sources will cite this as being part of the distal convoluted tubule. Other sources will say this particular reach of tubing is part of the collecting duct. Really, it's at the transition between the two. That's why there's disagreement about the name. But based on the physiology of what's happening at this spot, most sources now say it's part of the DCT. So I just wanted to acknowledge that if you run into that difference in terminology and you're like, wait, what? There is some disagreement about which part this is. And because of that, I'm not gonna like make you be super specific about it because it's not fair. If the scientific community doesn't agree about it, then I don't expect you to reach a conclusion either, but most evidence says DCT. Okay, so I'm gonna zoom in a little bit more, move that down. So what you'll notice, and this is a good skill to get in the habit of in general, is when you have a tube, sometimes the spacing of the cells will be not uniform around the tube, and that's true here. So we can see that the internuclear distance is pretty wide in some areas. But then once you get close to the juxtaglomerular cells and close to the side of this tube that's facing the corpuscle, we get lots of nuclei very close together. So they're touching each other in some cases. So I would say the macula densa starts at around here and it ends around there. Sometimes it's narrower, and sometimes the cells of the macula densa are actually so squished into each other that the nuclei of the cells end up being a little bit vertically elongated, but not always. So this is not true in this case. But regardless, these cells are not just functioning as a wall, they actually have a special job. Did I have somebody have a question? I heard a mic turn on. No? Okay, just make sure you're muted. Alrighty, so let's talk about this, which is the macula densa. And like always, I like to make sure that I'm being careful about defining the Latin and Greek roots because it helps you use your vocabulary knowledge more powerfully. Macula means spot. That's all. And densa means dense. So macula densa just means dense spot, which is referring to the fact that the cells are more densely populated in this spot on the tube wall. So sometimes things have fun, metaphorical, or fancy names, and sometimes things have very literal names, but they sound better if you say them in Latin, so we do. So that's the deal with that. So, dense spot, what does it do? Well, if we know, oh no, let's try and move this back, there we go. If we know that there's filtrate flowing through this thingy, and we know that it's end of DCT-ish by location, then what we will note is that we should see fairly low sodium, but still some passing through this, because urine is always going to be containing at least some amount of sodium. So sodium flow and the amount of sodium being detected as the filtrate passes through the tube is a proxy for measuring blood flow, because ultimately all this sodium comes from the blood, because our corpuscle is filtering blood, and a plasma-like substance enters. We call that filtrate. So if it's ultimately derived from the blood, then the amount of sodium and its fluctuations are going to be a proxy for how much blood we're filtering and how fast. So that's what the macula densa cells are sensing. They're not sensing blood flow directly. They're sensing sodium and using it as a proxy for detection of glomerular filtration and glomerular blood flow. So when glomerular blood flow and filtration are normal, we should see, a baseline amount of sodium, and this is going to be detected, and the juxtaglomerular cells, which I'll outline, but they're a little bit hard to see here. Why do I keep choosing pink? That's like the worst color for contrast purposes. Okay, that's better. So JG stands for juxtaglomerular. And I'll talk about that word in a moment because it's important to keep straight with a different word that is also juxta. So these are cells that live between the macula densa and the glomerulus at the the vascular pole. And then we can't necessarily be sure which arteriole is which, but pretty safe to say that this is where the afferent arteriole is plugging in to the glomerulus. And then it's probably related to this blood vessel, because I can see another portion of it here. But that's my best estimate based on what I see in my experience. So you have to kind of mentally connect to the dots for yourself. So that's probably the afferent arteriole. And I can also see that there's some smooth muscle cells around this particular point. So I can be pretty confident that that's the afferent arteriole. And then over here, I see another chunk, which is probably the efferent. So remember, afferent means blood's coming in, efferent means blood's leaving. And then we have the juxtaglomerular cells, which do a couple different things. One of them is secrete renin. So if we see a sudden and precipitous drop in sodium flow, like it goes from baseline to super low, what that implies is that filtration has taken a nosedive for some reason. Because in order for sodium to be passing through that tube and the filtrate to be filtered and dealt with properly, we should always see some amount of sodium in the filtrate in that tube. If that drops, that is noticed by the juxtaglomerular cells. And they're like, alert, alert, low blood flow. And in response, they secrete renin, or are thought to. So there's overwhelming evidence to suggest that these are the cells that secrete renin. So one way to test for that is to take some out and culture them so that you only have juxtaglomerular cells in your sample, which means that you can be sure of what they do, and then expose them to different concentrations of sodium and see what they do. And so if you do that, it turns out if you lower the sodium significantly, they just start releasing renin. So that's pretty compelling evidence. But remember, the renin-angiotensin-aldosterone system also includes erythropoietin, which is that one that acts on the bone marrow to cause erythrocyte production. And nobody's actually sure exactly which cells make that. So there's speculation and some medium evidence that the juxtaglomerular cells release that too. Also, why wouldn't they? But nobody knows that for sure. So we're just going to go EPO question mark. Probably, maybe, not sure though. So we've talked about the renin-angiotensin-aldosterone system a lot. So erythropoietin, wherever it's coming from in the kidney, somewhere, maybe here, is going to go to the red bone marrow and stimulate erythropoiesis, the synthesis of red blood cells. And then the renin is going to convert angiotensinogen from the liver into angiotensin 1, which kicks off that cascade of conversions that ultimately gets us to angiotensin 2, and then the secretion of aldosterone and ADH, as well as stimulating you to be really thirsty. So if flow is normal at some baseline, the JG cells will be quiet. If it drops and homeostasis is negatively disturbed, that's going to trigger the release of renin. So it's not necessarily the case that they're being suppressed from releasing renin that they want to release, but rather they have renin stored up, and if there's a drop away from that baseline, then they can release it at that time. And some of it also appears to be synthesized on the fly. It's not that hard to make as proteins go. It's not huge, so you can make some of it to order as well. But that's kind of the deal with the juxtaglomerular apparatus.
Please don't, I'm trying to.
So now that we've done that.
I have a quick question.
Yeah, go for it.
Is the renin getting released in response to the macula densa sensing the change in sodium, or is it? Okay, thank you.
Yeah, so it just sits there and pays attention to sodium only. And then if the sodium passing by it drops, then they're like, oh no, something's wrong. We should release some renin. It seems weird, like why wouldn't you detect blood flow directly as opposed to detecting this other thing? But if we think about the systems in place here, and we remember that the pressure difference and the diameter difference between the afferent and efferent arterioles is going to keep the glomerulus constantly pressurized enough to have a normal GFR. That already puts us in a position where the blood flow and pressure in the kidney is not normal. It's controlled in some way, and therefore we need to look at other things as indicators of flow, because blood pressure or the stretchiness of those vessels is not going to give us as much information in that context. So that's the deal there. Okay, so I'm going to pause for a moment. So now what we're going to do is zoom out a tad bit. Oh, look at all of those corpuscles. So you can see that even at low magnification, oh no, even at low magnification, they're very conspicuous. So like, ta-da. So you'll see that they're very conspicuous, which helps you establish the fact that you're in the cortex. And then if we move over a little bit, this is not my favorite view of the medulla that I've ever seen, because it's more cross-sectional, which makes your lives harder. But you definitely can see that, like, right about where this blue line is, there's a transition from places where you see corpuscles and fuzzy tubes and different organization, and now we're over here in like weird cross-section land. No corpuscles to speak of, just a bunch of tubes, including this very large interlobular artery. So I'm going to pivot to a different slide just so that the medulla area looks a little bit more normal, and we can look at tubes on there. Okay, so this is the one that I like the most. Also, this is a transverse plane section of the kidney. So instead of cutting it in half along its long axis, this is taking a kidney, which, you know, is a little bean shape, sitting vertically upright against your back body wall, and slicing it in the transverse plane. So as though you were being bisected your top half from your bottom half, right? So that's what this is. Due to this, you can see some pretty cool features of this thing. So I'll point those out. First of all, it's really easy to see where the cortex ends and the medulla begins. Not only is there an obvious color difference, but you can also see little corpuscles, and you can also see this transition and see all these tubes. And then we can also see the tip of the renal pyramid is called the renal papilla. Remember, papilla means nipple, so lots of different things in anatomy and physiology are called the something nipple because anatomists are just obsessed with nipples, I guess. But this is another one. So this is the renal papilla. There is one per renal pyramid. So it's just that tip out of which newly formed urine trickles. And then we've got, so it just means kidney nipple. Aha. So just the tip there. So I'm going to zoom in on the cortex and show you the DCT versus PCT stuff, but real quick, I do want to just zoom in on this area here because there's another thing you can see that is pretty cool that you can't see in a whole lot of other kidney slides. So let's wait for that to buffer. So although it's not visible here, because you can't see where this space goes to, you can see the space. And if we zoom even more in on it, we can take a little look at the epithelium and make some observations about it based on what the structure is. So this is always a tricky epithelial type. It's probably the hardest one that you encounter in 241. And that's because transitional epithelium, which is what this is, bears a striking resemblance to stratified squamous epithelium if you're a beginner and if you don't pay close attention. So here's how to tell the difference. And this is especially a sort of persnickety, tricky little guy, because when transitional epithelium is stretched due to the space it's lining being full of urine, which this looks like it was at the time of collection of this specimen, if it's stretched, the apical cells look like they're stretched out, and unfortunately creates a resemblance to squamous cells. Here's how to tell the difference though. If we look very closely, what we will see is that, sorry, I'm trying to find my power cord. There it is. Perfect. So the deal with distinguishing transitional epithelium, also called urothelium, from stratified squamous, which this is not, is to note the cell shape. So in stratified squamous epithelium, the basal cells look pretty round. And then the further up you go, starting about in the middle of the tissue, if you look at a stratified squamous epithelium, the cells start to flatten out. and that is most visible by observing their nuclear shape. So you don't see that here. The nuclei are more or less nice and round. They might be a little oval in some places, but they're not adopting an appearance that looks like this as they reach the surface. So even right up to the very top, you still see a pretty round nucleus here and here. It's only the very top ones in this case that are stretched and therefore accidentally look squamous. So in relaxed urothelium, these cells at the top, which are called umbrella cells, you can even tell that they're a different color. In a relaxed urothelium, the umbrella cells have kind of this domed shape because they've been allowed to elastically recoil to their original shape, which is capable of being stretched, but is not currently stretched. It's like a scrunchie. So your scrunchie only has ruffles when it is relaxed. If you stretch it out to the max, the fabric is no longer wrinkled. So in this case, we have some stretched umbrella cells, which makes their task a little bit harder, but you can pay attention to the nuclear morphology, and that should fill in the rest of the confusion. Also, if you look at this, and then you also look at where it is in the inset, or if I zoom out a little bit and you have some additional context, so you can see some papillary ducts here, and you can see this is the minor calyx, which is the first urinary space that the urine drains into. We know that transitional epithelium is specific to the urinary system. It's specialized to stretch because it has to accommodate large volume changes multiple times per day, and that it's lining all of the spaces from the minor calyx all the way through the ureter, into the bladder, and into the urethra. So we know it starts here. So if you see a renal papilla and you see a minor calyx, there should be no question about what this is. It's always going to be transitional epithelium, regardless of its morphological appearance, stretched or not stretched, because that's the only thing that's there. So other things that we can see on this slide that are of interest. One of the confusing things about the kidney that students have a hard time with, especially due to the way that the book illustrates it, is the idea of what a renal sinus is. So notice how the tissue stops at this margin, and then we have this space that is filled with adipose tissue. So you can kind of see the cell membranes here. It's not empty space. So adipose tissue and small amounts of connective tissue are what is in the renal sinus. It's the part of the kidney in the hilus or hilum that is not kidney tissue. It is devoid of kidney tissue, and that space is instead filled with fat, and it's used to anchor the tubes that are coming and going from the kidney at that location. So for any organ, the hilus or hilum is literally just the area on the organ where arteries are entering, veins are leaving, nerves are entering and leaving, lymphatics as well, and in the case of the kidney, also the renal pelvis and then the ureter. So a hilum is just a place where tubes come and go. It's not specific to any one organ either. And then renal sinus is full of fat. So adipose tissue represents the renal sinus. All right, let's go look at some tubes. So I'm going to go here, and I see lots of renal corpuscles, which means I know I'm in the cortex. And because I know I'm in the cortex, and I know what things are in the cortex, which is corpuscles, PCTs, DCTs, and then there are collecting ducts as well, which are organized into what are called medullary rays. Let's label a few. So see these kind of streaks of longitudinal tubes that are positioned amongst and in between the little clusters of corpuscles. those are all medullary rays. So rays referring to their sort of ray-like appearance, and medullary not because they're in the medulla, but because they are carrying pee down to the medulla, having collected it from all of the nephrons associated with all of these little corpuscles. So let's go look closely at what all that looks like. I'm trying to find a good spot. Well, I guess they're all kind of the same, so I'll just zoom in and we'll go from there. The only other thing I want to point out before I zoom in is, in addition to the medullary rays, which you can see from this low magnification, you should also be able to see that, like, if we just go ahead and outline the corpuscles, they're in like little clusters. They're linear, and they're pointing ray-like as well down into the medulla. So this is something that you would only be able to notice if you were zoomed out and had enough context to see it. I'm pointing it out because this helps conceptually connect the blood path through the kidney. So every single corpuscle has an afferent arteriole and an efferent arteriole, right? And those are both carrying oxygen-rich blood into and out of the glomerulus. That part's for filtration, so it's kept at a higher pressure ordinarily so that we can make filtrate. And all of the afferent arterioles arise from cortical radiate arteries, which are arteries that are positioned in the renal cortex, and they are radiate because they radiate away from the arcuate artery that lines the edge between the cortex and the medulla. You can actually see some arcuate arteries and veins here and here. So you can think of the corpuscles and specifically the corpuscles and their glomeruli as being like little fruits hanging off a tree, and the trunk of that tree is the cortical radiate artery. So we have like a little stalk of artery with little fruits coming off of it, and those are the glomeruli. So there's a nice illustration of that as well in your book that kind of represents that a little more clearly, but I'm just showing you that what is illustrated in your book can also be seen in histological section. And I think it's always nice to have it proved to you visually that the cutesy drawing and what they're claiming about a structure is actually true based on observation. And so we have the opportunity to look at that here. So now I'm going to go a scannin and just look for an area where I especially like the PCTs and DCTs. Move this pointer over here. I don't want that. Ooh. Remember I was saying in the previous portion of the video that you can always see either the vascular pole or the urinary pole in varying degrees, just depending on your angle of your slice. Pixely. So here's a place where, although we can't see the macula densa or the juxtaglomerular apparatus, we can really clearly see the afferent arteriole. Plug in right into this glomerulus, which is nice to be able to see. So one thing I recommend you do to prepare for the practical is to just go ahead and, once you've used your atlas and gotten familiar with the labeled images in your atlas, and you're starting to feel a little bit more comfortable, do what I'm doing. Don't use a little sidebar, whoa, as a crutch. Just cruise around and see what there is to see. You're on a little journey, taking a walk through a tissue, trying to discover stuff. And sometimes, like that situation I just showed you, you'll make some interesting discoveries that actually prove the concept that we're trying to illustrate. Here's another one. See that? Haven't gotten to any good urinary poles yet, but I'm hoping for one. I'm hoping to find one on the way to my area with lots of nice DCTs and PCTs. Well, no such luck so far, but I did find a piece of cortical tissue that I like, which has nice examples of each kind of tube. So we'll zoom in from there. Okay, so the difficulty of this tissue is that where formerly you were having to look at an epithelium and tell if it was cuboidal or something else. So you're like, oh, it's either simple cuboidal or it's stratified cuboidal, but probably not because that's rare, or it's another kind of thing. Now you have to be able to look at tubes that are all made of simple cuboidal epithelium and still tell them apart based on other characteristics besides the fact of their being cuboidal. So training wheels are well and truly off. You are in varsity level histology. Now, fortunately, there's other stuff about these tubes besides the epithelial type that make it actually not that hard to figure out what you're looking at. You just have to know the tricks of the trade, and that is what I'm here to tell you. So first things first, I do want to point out the very nice macula densa that we're able to see on this particular corpuscle. So see these cells here that are very, very densely packed, and you can tell by their nuclei being densely packed, and then they're more uniformly spaced out the rest. That's another example of the macula densa. But that's not the point of why we're here. We stopped here because we had an opportunity to look at some nicely differentiated proximal and distal convoluted tubules. And it's always handy if you're trying to distinguish between two things, if you can find a place where they're right next to each other, so you have them both in front of you available for comparison. So that's what I was looking for. So let's first tackle PCT. So the PCT cuboidal epithelial cells have abundant microvilli, and that's because if you want to exchange stuff and you're a cell, you can only exchange things as much surface area as you have. So you have a limited ability to exchange, and that is the surface of you, the cell, that is facing the lumen, so the apical surface. And there's a limited amount of cell membrane real estate there to put transporters in. So you kind of reach this theoretical maximum where if you have so many transporters that the membrane is like all transporters and very little phospholipids, it won't stay together. But you need lots of transport to happen. So just easy solution is if you make your apical surface wavy and have lots of folds, you can stick more transporters in that membrane and have more exchange for the same tube size. So that's the idea. So that fact and the fact of the transport activities that go on inside of the PCT, specifically as it relates to protons, causes the PCT cells to stain very on the eosinophilic side, meaning pinkish, reddish, orange shifted. Because eosin, that stain, is that color, and it likes to stick to certain stuff. So microvilli make a really messy apical surface of a epithelium, meaning the apical surface is the side of the epithelial cells that face the lumen. And if there's microvilli or cilia present, the apical surface will appear ragged and messy and not clean and uniform because there's little projections that the knife that we cut the tissue with has to pass through on the way. And so what you get is this messy, disorganized apical surface and lumen appearance. And then the stain, eosin tends to preferentially stain PCT cells. So we're looking for tubes with a messy, disorganized apical surface due to the brush border, and tubes with cells that are pretty pinkish red. So based on that description, what you'll find is there's a lot of tubes in this field of view that fit that bill. So let's start circling some. And I'm not going to circle every single one, but just, I'm trying to illustrate a point here so that you have a really nice frame of reference. You can see that that kind of tube is literally just all over the place. So there's another one over here and here, etc. So the other thing you'll notice is that if you pay attention to the nuclei in the cells themselves, they're kind of sporadically positioned and haphazardly arranged. So it's not the case that you see a ratio of one nucleus per cell and all the nuclei are in a nice little row. Instead, you see like stuff like this, where there's like nucleus and then cytoplasm, maybe a nucleus, and then lots more cytoplasm, and then another nucleus. So the uneven spacing of the nuclei is another sort of hallmark identification feature of proximal convoluted tubules. Oops, well, I can't undo that. We can see nephron loops histologically. I will show them to you after this, but nephron loops, just as a reminder, there we go, are going to be positioned in the renal medulla. So we're going to have to go to the opposite side of the tissue from here to do it. So now we're going to pivot to finding distal convoluted tubules. So because so much transport has occurred, by the time the filtrate gets to the distal convoluted tubule, remember it's gone through the PCT and experienced a bunch of reabsorption and secretion, and it's also passed through the nephron loop and had lots and lots of salt and water taken out of it. So because of that, the DCTs do not need to have the same brush border as the PCTs do. So what we're going to look for, and I'm going to circle them in a different color. Let's do yellow. What we're looking for are, again, simple cuboidal epithelial cells, but they have a much tidier apical surface. So you can actually see the delineation between the apical surface cells and the lumen lots more clearly. There isn't that messiness. And the cells that make up that tube don't stain as brightly, especially not their cytoplasm. They're not really that eosinophilic. They're just kind of medium. So you've got this light colored medium pink cytoplasm. And you can also see that, with some exceptions, because no rule in histology is 100%, but the nuclei that make up the DCT cell are mostly evenly spaced, especially as compared to the proximal convoluted tubules. So you're still looking for simple cuboidal epithelium, but you are looking for tubes that are much tidier looking and not as bright pink, with uniform nuclear spacing and a very clean edge. So let's circle some of those. Darn it, keep moving this. I need to do that. Well, I'm going to make this line up a little better. There we go. So there's one. There's one. This is not my favorite one on earth, but it's still one. Here's one. Here's one. There's one. There's one. And this is one as well. So let's make ourselves a key. And then finally, remember that the DCTs all plug into collecting ducts. So multiple collecting ducts, or excuse me, multiple distal convoluted tubules are going to empty into the same collecting duct, and that collecting duct is going to take all of the urine from multiple distal convoluted tubules, therefore multiple nephrons, and take that down into the medulla so it can pass out. Oops, I want yellow. So this is also a DCT. And then we're going to add a third color. So what you'll notice is that the DCTs have that appearance that I talked about, but there's another epithelial lined tube in the vicinity that resembles a DCT, but their structural orientation, like the physical way that they point, is more uniform than the DCTs. So you can see for all of these, we're kind of, looking at tubes that are cross-sectional, semi-cross-sectional. So here's cross-sectional almost entirely. Here's semi-cross-sectional or oblique sections, but they're kind of pointed in all different directions. And then you get these other tubes which look similar, but they're definitely pointing away from the surface and down towards the medulla. Oop. So those are going to be collecting ducts. So if I pick one of those to show you, I'll make sure I pick one that is accompanied by other branches of collecting ducts, and you'll be able to see them sort of heading into the renal medulla and being part of a medullary ray, which collecting ducts are part of the medullary rays, which are those ray-like swaths of tubes that I showed you from more zoomed out. Medullary rays include collecting ducts and also vascular components that are transitioning between the cortex and the medulla. So things like cortical radiate arteries and or veins are also going to travel in parallel with collecting ducts. So now let's go look at the renal medulla. So if I zoom out, we can see that there's this ray-like structure right here. So let's zoom in and suss out what this is. Pretty easy to tell. I'm seeing squamous endothelium and smooth muscle underneath. This is going to be one of those cortical radiate arteries. And then if we zoom out a little, we can see that there are multiple corpuscles kind of clustered around it. So the afferent arterioles leading to these corpuscles are going to be coming from this cortical radiate artery. And in some cases, you can actually, if you look closely, make out the point at which the afferent arteriole breaks off from the cortical radiate artery and heads towards a particular corpuscle. So there's one. So if this is the branch, here's a little stem. That's probably going to this guy. So again, the corpuscles are going to be the fruit. And then if we look over here, we can see another branch. And we're not sure where that's going, but given the fact that it's nearest to this one, it's probably headed that way. So if you look really closely and you think about what you know about the gross anatomy of the kidney, and then you apply what you know to the microscopic anatomy, there's actually a lot to see in addition to the various tubes and such. As long as you kind of logic your way through it and be like, okay, well, the afferent arteriole has to be connected to some artery. Which one is it? It's the cortical radiate. So if I find a cortical radiate artery, which is going to be an artery that's running through the cortex down towards the medulla, and it's relatively straight. And I should also be able to see which corpuscles I think are associated with that particular cortical radiate artery. So in addition to visual identification and just kind of sussing out, visual differences, it's always a really good idea to apply logic and larger order structural learning to what you're learning, because that helps make sense of all of it. Okay, you can also see the collecting ducts as they descend within the medullary rays. So I see lots of PCTs here, so we're still in the cortex. We haven't made it into the medulla yet, but this collecting duct and this one too, the transition from cuboidal to columnar cells has already begun. By the time this collecting duct gets to the area next to the renal papilla, so the very tip, these cells are going to have changed to be fully columnar and also lighter staining. So I'm just going to follow this medullary ray down because they all point to the renal medulla. And here we can see medullary rays going down in there. So I'm going to pick just an area that I like the looks of. I like areas where they're pretty straight like this because it's just easier to tell things apart. Let's actually zoom out just a little bit. Okay, so first things first, it's helpful to make just a list for yourself of okay, I know I'm in the renal medulla. What kinds of tubes do I expect to see here? I'm just going to make myself a little area to draw, like so. So I'm going to make my list called medullary tubes. Which ones are there? Well, there are collecting ducts because the urine has to escape somehow. But I also know that my nephron loops poke down into this thing. So that implies that I'm going to see thin descending limbs of the loop of Henle, and also thick ascending limbs. And depending on how good of a slice we got, also elements of the vasa recta, which is a ladder-like capillary bed that surrounds the nephron loop. So let me just draw a schematic of that really quick, just so you have some point of reference. So, if this here, I can see it's eosinophilic and cuboidal, but we're in the medulla, not the cortex, so it's not a PCT. The thick ascending limb is going to look similar to a long version of the PCT, but it's in the medulla, so it's not the PCT, therefore it must be the thick ascending limb. And then right here, we've got a squamous lined space, and I'll outline these more in a minute. But if we know that the peritubular capillaries of the vasa recta wrap around these tubes so that the water that's moving out and the salt that's moving out has somewhere to go, then I'm going to expect the vasa recta to look something like this, where we have descent into a ladder-like shape that provides an area in the middle where gas exchange and other stuff exchange is going to occur. And then we have the venous end over here, so that all of the happenings in the thin descending limb, outward movement of water, for example, that's going to end up in this blood. and the outward movement of salt is going to move salt into the interstitial space, but as the salt transport continues all the time, over time, new salt will be replaced by old salt, or old salt will be replaced by new salt, excuse me, and we'll get sodium chloride back into the blood this way as well. So that's what the vasa recta looks like. So if you can logically think about its shape, it'll be easier to find. So again, looking carefully at things like cell color, nucleus spacing, apical surface, we can find our thick ascending limbs. Oops, thinner dot. There we go. Here I see one, there's one, there's one. And remember, these are all packed closely together into the renal medulla. So you might see slices of one ascending limb and then another, and they're all kind of smushed into each other. That's fine. As long as you pay attention to the cell morphology making up the tube, you should be able to tell. And so I'm going to say these are green. Let's do collecting ducts in this aqua color. So here's like a big long one. Whee! And another. And then we get like kind of, we got the middle of the tube here, and now we're getting the edge of the tube, so the cells appear to converge, but still a tube. So here's another one. There's another one up there, etc. And that leaves us with thin limb versus vasa recta. So your problem with these is they're both lined with simple squamous epithelium because blood vessels are, and the thin descending limb also is. So how do you be sure that you're looking at one or the other? Well, a helpful hint, and one that I'll use too, is I'll just look at it and see if I can see any other things in it and whether or not I can see erythrocytes in it. So do that. Let's go look for some stuff. Wish I could twirl this, but unfortunately I can't. So just going to give myself a very little dot. So if I look at certain tubes, like I can see erythrocytes in this tube. It's very thin and it's running vertically. And it's also next to a thick ascending limb and this guy, which is a thin descending limb. So lined with squamous, very, very simple, but no erythrocytes to speak of. And it's right up against a thick ascending limb. So that can make me confident that this one that I'm going to outline in orange is going to be a thin limb. So again, definitely training wheels off for distinguishing between two different things that are made of the same epithelium. But if you pay close attention, you think logically, you apply what you know about the physiology of the system and sort of logic it out, you can get yourself the rest of the way to mastery. So here's a really good vasa recta situation. Which is pretty cool. And then if you just kind of scan, you can see other stuff as well, like, oh, here's a turnaround point. That's neat. So that's going to be the place at which the thin descending limb, which is probably this one maybe, is turning around to become the thick ascending limb, but you can't necessarily see exactly which tubes those are associated with. And then before we stop this particular recording, let's cruise on down to the very edge. So what you'll start to see is the epithelium lining the collecting ducts is becoming more and more columnar. And if I just continue on down, it starts to be conspicuously columnar, right? So like all of these guys that are very obvious now and very columnar epithelium-ish. So as the epithelium of the collecting duct approaches the end, we start to call these papillary ducts. They're still collecting ducts. It's not that it's a different tube. It's just when we can see that the tube is close to its exit and that its epithelium has changed significantly, we start to call it a papillary duct because we know that we're near the renal papilla. So which means that if we continue following these things down, we should get to, oh, voila, the renal papilla. And you'll notice that surrounding this very, very distal, deep medulla, there's pretty much just stromal cells. There's not really any thin or thick limbs anymore. We've just got papillary ducts anchored in stromal tissue because they're literally just here to drain urine into this minor calyx space. Adios, have a nice night. And then we can see minor calyx space, transitional epithelium, a little bit of connective tissue, and then the fat of the renal sinus. So that concludes